Hi, so my name is Isabella, and I'm going to be going over my article with you today. I'm going to go ahead and share my screen so you can go ahead and take a look at my presentation. So for today, my article was Canine Tumor Cell Line Panel, a resource of drug discovery in comparative oncology and translational medicine. So just a brief overview, the researcher's gap of knowledge in this article is trying to understand cancer and both its cellular formation and response to chemotherapy between both human and canine and how comparative it really is to each other. They're looking at both the phenotypic and genomic values and cell data that there is, since only studies have been done on human cancer cell lines. The figure that I chose depicts the drug sensitivity and the 6 different types of chemotherapy, which are listed here. So for these, we have cisplatin, paclitaxel, lomustine, vinblastine, carboplatin, and doxorubicin. So again, the name of the article is the Flint Animal Cancer Center Canine Tumor Cell Line Panel. This article serves to look at the cell tumor lines with the different types of chemotherapy that are available, whether they are meant for canine or they're considered extra label, which means they're not technically meant for canines, only for human, because there were not enough studies done to actually make it a veterinary medicine prescription. Experimental methods that were used were the canine FACC panel via Resazarin assay. And they compared it to the human panel that was used for cancer tumor cell lines. So the NCI 60 cancer cell line panel is a group of 60 human cancer cells that was used by the National Cancer Institute for screening, and it helps to detect anti-cancer activity. So from there, that's when they decided to do the canine FACC panel, which is the Flint Animal Care Center panel. And again, it is a screening test that looks at the early stage compounds in a group of cell lines to identify anti-proliferative activity, as well as looking at larger amounts of cell lines to find predictive genomic biomarkers of response. So with that, the independent variable in this study was the anti-neoplastic drugs, which are chemotherapy, and the dependent was the log GI 50 mean, which is the drug sensitivity to cancer tissues. Negative control, again, drug sensitivity, and the positive control was the canine tumor cell lines. So with that being said, just to go a little bit into detail, the anti-neoplastic drugs or the chemotherapy is the independent variable in this study. So with that being said, the reactions are actually the ones that are known to be the dependent variable. So the drugs are known to affect that dependent variable because they're ultimately affecting the tumor cell lines for both the canine and humans. With the graph, we can see that there's significant differences with the drug sensitivity between animals and canines. So one of them was carboplatin, in which it showed a value of negative 3.8 for humans and negative 5.5 for canine. And paclitaxel, which showed a value of negative 6.9 for humans and negative 9.6 for canine. So when we go back up to my graph, we will be able to see the very distinct differences. So let me see, paclitaxel is right here. So we can see the human cell line was, again, between the -9 and -8 number. And for canine, it was actually by the -7, closer to the -7, but still between that -7, -6. Where we see no significant difference is going to be the cisplatin, which is between human and canine. So the human is between the -6 and -5. I want to say it's about -5.4, and canine is -5.5. So it just shows that there's no really significant difference between chemotherapy and the drug sensitivity for cisplatin, but for the other ones, there was a significant difference. I don't believe there are any faults when looking at the graph, other than it being a little difficult to understand when first looking at it. I did have to read the paragraph multiple times to try to understand it more. Something that could ultimately affect the results, though, that the researchers may or may not have thought of was how far along the cancer was, whether that tumor was once resected and it grew back, and also are the canines and humans within the same stages of cancer. So that would be a really big thing, as well as how many rounds of chemotherapy did they each go through. Overall, the color coding of the graph did help. I really did appreciate that from them because it helped me to really differentiate the different types of chemotherapy there was. The figure description itself was fine. It explained what the graph was used for. And the only thing again was the log that was used towards the bottom of the graph for the dependent variable. That was a little difficult to understand, but again, it was easy to get through once I read through the article multiple times. And yeah, that is my article for today.